to lead off. Lucian is going to lead off on this one. Just going to talk about standards and so on. I just want to add something to that in case Lucian doesn't mention it. But the whole idea of exchange formats and so on is really crucial to reproducibility. I mean, this is hosted by the Reproducibility Center, so I should mention that. The bulk, if you went back in time 15 years ago, you'd find that pretty much 100% of all computational models, simulation models, could not be reproduced from the literature. Now about 50% can, and that is pretty much down to because people are using standards like SBML. Because once it's in SBML, that's a description of the model. It's a very precise description of the model. And then you just need to find a tool that can read it and then run it, and that's it. And the other advantage is the model becomes highly reusable, because now it describes the actual model in detail, and you can just pick pieces out of it or whatever and reuse it somewhere else. Whereas if you publish your model in raw MATLAB, for example, that's much harder. Plus, when it's in MATLAB, you can only really run it in MATLAB unless you take the effort to translate it to something else. Anyway, I just wanted to mention that this whole standard stuff is really important for reproducibility. Okay, Lucian, I'll hand it over to you.
All right. Thank you. So this is, excuse me, the Colab notebook that I have in the session 4. It's the only thing there. Again, I just have the same introduction here. Don't forget to restart the runtime. You can run this just to sort of make sure that everything is running. So, which it is on my computer. So, as Herbert mentioned, SBML is a standard format for models. And Tellurium uses SBML sort of behind the scenes. We've talked about Antimony as sort of the easy to write front end for that. But if you, well, here, you know what, I'm going to add another, we'll just do this. So species S1 equals 3, right? We just have a really basic thing. Oops, here. R equals, and then R simulate plot, right? Here's our very simple, like, we have a species, nothing is going on with it. If you want to get that, if you want to see what the model is in SBML, you can say get SBML. Oops, no. I think you can just do get SBML, right? Yeah. And here is that, and we'll make it look slightly nicer. There. So this is a complete, if very simple, SBML model. And you can see the little things in it. It's XML based. We have a compartment that's a default compartment. And we have our S1 species in it that just has an initial value of 3. And that's it. That's the only thing that happens here. As models get more complicated, that gets harder and harder to read, which is why we use Antimony instead of SBML raw. Excuse me. So over the years, since the early 2000s, a place called BioModels has been collecting SBML versions of models from papers. Often they will go take a published paper and create the SBML model from it. Sometimes the authors do that themselves. Sometimes the BioModels curators work in concert with the authors of the papers. Sometimes this is, like, usually this is a very complicated process. There's not enough information in the paper to fully recapitulate the model, so you have to go back and fill in all the blanks. But in the end, if you go to biomodels.net, they now have over 1,000 models that have been manually curated, that all have been used to recreate at least one figure from the paper that they come from. And then that makes them, as Herbert said, reusable in other contexts. You can recapitulate the information from that paper itself, and then you can extend it, you can change things, you can analyze things in different ways. You can do parameter fitting, you can do metabolic control analysis, which we'll talk about later, other things like that. The non-curated branch here is has a lot of models that are in some stage of curation. Many of them are basically already and just sort of are awaiting a curator's stamp of approval. Some of them are sort of first drafts of models, so you never know what you're going to get in the non-curated branch, but there are many good models there anyway. And the auto-generated branch has models that are generated from big data, so they tend to be like really like yeast metabolism for this particular species, yeast metabolism for this other particular species, stuff like that. And generally, the auto-generated ones do not have kinetics in them; they are just sort of structural. We know that metabolism works by converting this to this, and we don't know the rate at which that happens. So, for purposes of this workshop, I'm going to show you a particular model, and then we'll sort of go through and analyze it a little bit more. So the model itself is BioModels 10 from MapK oscillation. So if we look here on BioModels, you'll see different files. There's a lot of sort of extra files here, but the main one here is at the top, the model file. This is what BioModels has worked on making for you, and will contain the SBML file. You can see in the curation tab, this is a figure from the paper that they recreated using that model. Let's see, anything in particular here. You can say see things were changed slightly over time, but again, the main things are this one and you can see the curation as the figure that someone made at some point. The paper itself is at this link, and in particular, the figure we want to reproduce is here. So this will jump right here, and you'll see, again, so from the curation tab, this looks the same as the other one did. So what we want to do is we want to sort of get Tellurium to the point where we could recreate this figure. And then we'll talk about figure B also, because you can't do that directly, but you can do it sort of indirectly, and that all can be worked out. So if you look at the figure, you see the time goes from 0 to 150. It has a couple species in it where the labels of what exactly is being produced is not stated. So we'll just go with the defaults, and we'll simulate from 0 to 150 and see what happens. This first line here, load S, is for load SBML. And this is the download link. If you go here to this download link right here, you can copy it. And that's what I did for this bit. I pasted it in there. That's exactly where I got that. So simulate it for 150, just like the plot, just like the figure, and then we look at it. And immediately we notice like there's some movement here, but it doesn't look the same. And can anyone guess what the issue is? Anyone? It's relatively straightforward. So we'll go back and look at this. And this says the time in minutes. So as you might guess, the time in the model, the native time units are in seconds. So this won't work as well as seconds. So we just take 150 times 60 and just do the same thing. And look, we have our oscillations. Now this figure has a lot more species in it than the figure in the paper. But if we look, if we're looking at this, you can see this first oscillation is sort of thick here and then thins out and goes a total of sort of 1, 2, 3, 4, 5, 6, 7 times. And if you look for something that matches, you'll see this gray line here is 1, 2, 3, 4, 5, 6, and then 7 behind this. And the gray line looks like it's mapk_pp. If we then look for what is coming up, like starts high, drops for a while, and then comes up to around 200 in between the other peaks, we can see like, oh, look, this brown line is doing that. That's just mapk. So it's basically what this figure is showing you is just MapK and MapK double phosphorylated. So based on those labels, I can then set the selections to that. I could also, as Herbert did, put them in here as an argument. But when we do this, we're just looking at time, MapK-PP and MapK, and indeed we get this sort of standard, the figure which we had from the paper. So that happens to be something that worked really easily. I picked chosen because I knew it would work. There are other figures that we know have been used to recreate these at some point, but that might be a little bit more difficult to actually reproduce the figures if you're working at them. So here we had to figure out like what the time scale was and which species in particular we were looking at, which was relatively straightforward. But it won't always be super obvious what happens in other cases. Now, as it happens, so the fact that there was some difficulty here, and there'd be, excuse me, take a drink. The fact that there was some difficulty here, and the fact that it will get even more complicated as you go on with more things, means that another standard has been introduced called SBML, or the Simulation Experiment Description Markup Language. So SBML is designed to encode basically this information. Like we're just looking at these particular outputs. This is our time period. We're going to 150 times 60 instead of 150. We're taking 1,000 points along the way. You can even encode information about like what the x-axis should be labeled, what the y-axis should be labeled, like, etc. So if someone has done that, then it's even easier to reproduce figures from the paper because that's already been done for you. In the early days of BioModels, SBML didn't even exist, and they've been sort of slow to sort of catch up on that. So most of the SBML, that is, every so often you will find some models in BioModels with some SBML in it. In general, it has not been even valid as SBML because there hasn't been a lot of tests for that. At the center, we've been working on improving that. So at this point, we have a lot of SBML that is at least valid, will produce some sort of simulation. Sometimes it'll be close, at least, to what's in the paper. Sometimes it's just sort of a base default simulation that you can work with to sort of bootstrap your way to the actual figure. So as noted here at the center, and there's our center URL, I actually went and curated this particular thing again. So whoever made the original model like made those figures, and what I tried to do is make both figure 2A and 2B. And 2B, as you will see from the figure or from the legend, B says cooperative inhibition with N equals 2, the activation of medicinal kinases by MapK-PP, and then it has a bunch of things that were changed. So K I or L or whatever that is was 18, K1 was 50, and then a bunch of others were 100, and then the couple V values were 1.25. It says the other kinetic parameters were the same as in A, and that was all just described in the paper in general. I don't even know. Yeah, here we go. So here's your original, like this was originally written just as a series of differential equations with particular parameter values here. Part of the translating it to SBML meant changing these differential equations into actual reactions, which made things a little bit more straightforward to read and to understand. So I actually went through, made the ability to make both figure 1 and figure 2 out of this, and I have them up at biosimulations.org, and this is not the best interface, but at least it works, and we're working on improving this also. So you can select figure 2A and look at it, and Yes. Okay, good. Here it is. You'll notice I was actually able to like even make one of the lines solid and the other line dotted, so it would match the figure even better. And if you go and select figure 2B and view that, oops. Click view here. There we go. This then is the other one with the dampened oscillations for both, where map K PP, which you can see by hovering your mouse over the thing, goes down a little bit. Map K goes up to roughly where it was before. So you could see the actual two figures here. Let's see here. So the other thing that this site has now is all of the information. Like this was simulated with Tellurium and stuff. So again, the advantage of storing things in SBML, the advantage of storing things in SystemsML is that you can run these things on a variety of simulators. I ran these on biosimulations with Tellurium, but I could run them with Coposi, I could run them with B-Cell, I could run them with a few other things as well. And if we go to the files, you can actually see all the things that I use to create it. And here is the SetML file. And I think I downloaded it. So yes, so there's a link here to download it. And in the collab thing, you can use wget to grab them. So I'm grabbing this SetML file, I'm grabbing the SBML file, and then I'm grabbing this other file called an OMEX file. So an OMEX file is just a zip archive of all the things in, let's see here, in here. So the OMEX file will have the SBML file, it'll have the SetML file, it'll have all these pictures, it'll have anything else that you want to stick in there to that you want to associate with your model. And the main thing that helps is because the SetML file needs at least one SBML file to work, you then have to sort of keep them together. And if you stick them in the same zip file, and in this case, an OMEX file, then they will always stay together and you don't have to worry about them getting out of sync with each other, or one getting lost, or one getting copied to a wrong directory or anything like that. So this one just grabs those from biosimulations, and then I will use this to look at them. So this is a quick, look at this here. So several years back at this point, it probably needs to be updated. Hirichoi and I sort of worked on something we call PhrasedML, and PhrasedML is sort of, PhrasedML is this SetML, what Antimony is to SBML. So Antimony is the human readable version of SBML. PhrasedML is similarly sort of the human readable version of SetML, the description of the things that are changed. So here we see we have the Kolodenko model that's just load this file. And then for Kolodenko B, it's the other model with n equals 2, with ki equals 18, k1 equals 50, a bunch of things, k2 equals 40, everything, a bunch of things equaling 100. And there's our v9 and v10 eating 1.25. And if you remember, that is basically all of these changes that the caption told us to change. The other thing you'll notice is that these two simulations go for different amounts of time. This first one goes to 150 and this one goes to 200. So I told it, let's move back here.
Sorry, Lucian.
Yeah,
can
absolutely.
I interrupt you a little bit?
Yep.
Sorry, Lucian, but for me, at least, I guess it's not a little bit clear how do you get the so I just asked if you can recapitulate a little bit the pipeline. So you run it first in let's say in locally use Tellurium and Antimonium, and then you dump it to a SetML file, and then you run it, or you have a repo a public repo of SBML?
Yes, that's a very good question. So, yeah, let me explain all the pieces. So locally, I've created a Python script that would recapitulate those figures. And then I had to specifically take all that, take the instructions that I made and translate them to SetML. I used PhrasedML to do that. I sort of like, this is the translation of the final version back into PhrasedML. So I wrote something like this, just like I might create a model in antimony to describe the process that I wanted it to go through. PhrasedML is a little old. I had to like actually edit the SetML directly a little bit because I know all the things that are going on. But basically, I created a SetML file. Then I wrapped up the SetML file and the SBML file together in an Omex file. And then I uploaded that Omex file to biosimulators. And this is a little bit awkward, but since you asked, I'll go ahead and show you how to do this. This has a lot of graphs, of which you need to worry about like 3 of them. So if we will look here, let's see. I have a lot of, so here in, so this is where I've been working on like updating all the BioModels and making good SetML for them. And so here is BioModels 10, the Omex file, and I think this is the most recent version, but if it's not, that'll be instructive also. So I have an Omex file that I created partly by hand, partly by using these Python things that I will then send to the project. It then realizes, oh, it's an SBML, it's using CVOD as the basic framework. Then it asks you to pick a simulation tool. So let's pick Copasi just to make it slightly different. Then I have to like call it something here, give it some text, and go Copasi, and then I can click run. So once I have an Omex file, then I can upload it here. And once something's been uploaded here, anyone else can then look at it if I give them this URL. So in fact, here I will copy this into chat. Chink. And so now that's a little complicated, but okay. Anyway, let's see. Okay, so this succeeded. Let's go to here. We'll select the file. Oh, and this version only had the auto-generated plot. So that is, so before I had curated that was what I had. And indeed, you see things slightly moving, but not much has happened. And it just goes from 0 to 10 because there wasn't anything at BioModels to tell in the BioModels files to tell me specifically that I needed to run it to 150 times 60. So it gives you the basic thing of just like, well, here's something which will hopefully be somewhat helpful.
Okay, but just one question. So the first one, the SBML of the first one that you're showing was the one that you dumped from the notebook that you were running. So the time was properly set. So
yes, the first one was one that I had made, and I
okay.
If I had uploaded a different one, then the time would be properly set, right? So that's whichever it was. This one probably. Nope. Oh, we will. Oh, sorry. Yeah. Okay. So this is just back several. I think. Oh, all right. Sorry. Okay, here's my run for that one. This is the one where I had uploaded the one with actually figure 2A in it and stuff. So if you were to go to a random one, so after we finished helping BioModels fix all their SBML, you'll get slightly better things, but you wouldn't get, well, for this particular one, since I curated it by hand, you'll get this one. Jin is also working on going through things and getting things so they actually match the figure. If not, then you'll get something that can start you off. Basically, it would be something more like what we started with. In this case, it only went to 10. And then you can sort of try to extrapolate from there. Does that help? Does that make more sense?
Yeah, thanks.
All right, sure, thanks. And anyone else, feel free to jump in with other questions as we go too. All right.
I guess there's just one thing. I don't know if you're going to cover this. Just to go over how somebody can, they've entered a model in Antimony, how they can get the SBML. I guess you did that, and how they can save it, and how they can load it from a file.
Right, that was what I did a little bit of here. Just
yeah, you got, okay, so you got
so
the get SBML.
We just
there.
printed it, just get SBML. But if you wanted to save to a file, there's a Tellurium.
Yeah.
Save to file.
File name.
Yeah, I think it's R.
No, it's file name first.
Oh, it's file name. Okay.
Yeah, from to, I think. Yeah.
Simple.xml and then R.
So that's how you would, save your SBML,
right.
if you after you've created your model.
Right. So now if I were to, here, let me do a new one. Te.load s simple.xml.
That's it. That's how you bring it back in.
Right.
Because you had
then you
R
would go
equals.
Here we'll print get antimony, which you can also get.
You don't need
Nope.
Don't need R.
No, here.
Yeah, R. There you go.
There.
Bingo. So you can move things, in and out quite easily
Right.
in SBML.
Right. So there we save this to a file. Our original antimony string was just this, and then the round tripped version has some comments in it just to help describe things. But yeah, that'll be the round trip. Yeah.
Yeah.
And you could take your SBML file, and then you could take simple.xml and give it to Kopasi if you were running Kopasi separately or anything else.
Yeah.
This was the creation of SBML was because people had a lot of different tools that could do different sorts of analyses on different models. And originally, if you wanted to do analysis A and then do analysis B, you had to completely create from scratch your model in each tool separately. And then, like, maybe you made a mistake. Maybe you typoed a 3 instead of a 3.0 or 3.1 or anything else. So once SBML became a standard sort of lingua franca for the model, then you could create your model in one tool, do your analysis, export it to SBML, and then you would import that same file into another simulator software analysis tool, and you would know that you had literally the exact same model as you had before. Similarly, with these BioModels, you load the model and you have exactly the same model that was used to recreate those figures from the paper.
The other advantage now of creating it in SBML is it can be uploaded to a repository. So you may have not noticed that I'm not sure if there are any MATLAB models on BioModels.
It was exclusively SBML for many years. They're trying to expand that a little bit now. But
the general trend is if you publish a model in MATLAB, it's unlikely to become available on one of the repositories.
Yeah.
So if you want your model to be out there in the world so people can use it, convert it to SBML, then submit it to BioModels. so they can have
Right.
it. In fact, there was a paper that came out earlier this year that showed if you do use these exchange formats, your paper is more likely to be cited. So
Yeah.
there's a
As
good
people
incentive.
actually use your thing, and we're like,
Yeah.
oh, let's extend science and
Yeah.
make things better.
I mean, a couple of months ago, we took almost a month. There was Michael involved. We almost took a month trying to convert a big MATLAB model into SBML. They're not, depending on the model, it can be quite difficult to,
Yeah.
reuse something like a MATLAB model.
Yeah. And in there, like these figure things are even harder. Like those are hard coded into
Yeah.
some specific things. Like if you wanted to run this out twice as long, you'd have to go find like where's the end simulation time, like where does it do anything, like, and all that is just
By the way,
buried
somebody
in
had the question,
coded things.
somebody had the question, how would I change the x-axis units here? Because it goes from 0 to 8,000, right? But the paper goes from 0 to
Right.
200 or whatever. I mean, one
And I-
possibility is to change the parameters so they're in minutes. The units are minutes rather than seconds.
Right. And you'll
Another
notice on this,
yeah,
I
how did
actually
you do
did
that?
that in the SBML. I just took the time and divided it by 60, and now the x-axis is like goes up to 150 like it should.
Okay, yeah. So you can, so the data that comes back from the simulation, just rescale the first column. So instead of
Yeah.
the seconds, it's now minutes.
And in fact, yeah, here we go. Where I did it here is right here. I could say I took the time and divided by 60. I'm plotting that versus MapKPP and MapK. So the format here is like I'm plotting this versus a list of things afterwards.
Right.
This part. So yeah, so this is sort of the human readable version of what was happening here. We have the two different simulations, one that goes just to 9,000, one that goes to 12,000. Again, that's in seconds, not minutes. And then and then we scale it back here, back to 60, but back to minutes by dividing the time by 60. You could change the model itself to work in minutes instead of seconds. That might be a little bit more awkward. You'd have to sort of know what you were doing.
Yeah.
Like all of your rates you'd have to change, all of your k values. I think the simplest thing might be to just change all your k values, but you'd have to make sure you got every last k value and every last,
and you didn't make a
everything
mistake.
else.
Yeah.
And make no mistakes. This is a lot simpler. Just like take the data, divide the output by 60, and then you've rescaled everything.
Yeah, that's the one.
Yeah. So we got our two different simulations. We have our two, what SBML called tasks, run sim0, this first simulation on Kolodenko, which was our base model. And then I'm running run sim1, which was a slightly longer one on Kolodenko B, which had all these values changed, and then plot the results. And let's, oh yeah, that was that. Okay. So when, and then as mentioned, I actually downloaded the actual file as an omics file, and Tellurium can execute it directly. So if you have an omics file, you don't have to go to biosimulations and stuff. You can just run it directly, and it does that. It's printing out all the results here for you, which oops. And printing everything. But here is the thing, and it even labels it figure 2A, MapK concentrations in nanomolar, and then in the x-axis even says time in minutes. So it is, you have a solid line, you have a dotted line, like everything is like we have recapitulated that particular figure. And then figure 2B, we have our two MapKPP, MapKs, MapK concentrations, time in minutes. And then this is the the next bit is like the Python code that Tellurium generated to do that for you, which you might be interested in if you wanted to like edit it or just sort of see what's going on, or if you had some question about like this omics file can do something for me, how can I do the same sort of thing? You could look at the Python code and hopefully get that sort of thing. And then this is the actual data that is also generated, because there were different reports. Here, let me show you that part too. Let's see. So here, there are also output reports, and on biosimulations, it'll give them to you as HDF5 files. Tellurium is not as complicated as giving you HDF5 files and just gives you CSV files. And you can actually, this function, execute combine archive. Oh, here it is. You can save the outputs as CSV and PNGs or PDFs, I think, into a particular output directory if you like. The plotting engine, I think there's only, I think matplotlib is your basic plotting engine, which that's the one that works more consistently, but if there's a problem, you can sometimes try to change that. So that's what that is. So that is the basics of what we can use. So the hope is that as we go forward, you'll be able to find not only SBML models for your models, but also SetML files and OMEX files for your models. So you can not only say I have the model itself, but also what you did with it, what the specifics of the simulation was that let you reproduce things. Are there any questions about any of that? If not, I will hand things over to Jin, who is going to talk about visualizing SBML networks.
Hello, everyone. My name is Jean. So I'm going to talk about a Python package to process and visualize SBML layout render. So as Lucian has introduced, SBML files are machine-readable representations of systems biology markup language. So it has informations of all the models like compartment, species, reactants, products, kinetic laws. So SBML level 3 has a layout render extension, so it can store the positions and render information of it. So layout describes the position sizes for different graphical objects, including compartment, species, directions. Render describe color and shape informations, like fill color, border color, line color, and shape of nodes. So first of all, let's see an example of visualization. So this example shows compartments can be in different colors, and node can be in different shapes. Aliens nodes are also supported. So aliens nodes are common nodes between or among different directions. This is useful for visualization of large-scale networks. And also border colors can be different. Reaction lines can be different colors. So SBML diagrams support PNG, JPG, PDF export of the figures. So you can see more information online on GitHub for a tutorial, introduction, or some examples. So this is a real example of glycolysis model. Joe was talking about the parameter fitting of this model this morning. And for visualization, you can see the reaction lines can be in basal curves, alias nodes in dashed lines, and bordering nodes in thicker lines here. Okay, so we think SBML diagrams is useful for process of the layout render information. So as Lucian has introduced, you can generate a SBML file from antimony by this r.getSBML. So you can load it to SBML diagrams and do an auto layout and draw, then you can have a figure with it. You can also use SBML diagrams to added an existed SBML file. So after load it, you can set it and then export it, and the information updated is saved in SBML file. So what SBML diagrams can get or added, including the following situations. So after you load an SBML file, you can get the layout and render information of it, or set or the layout and render information. So layout, for example, compartment position, node size, reaction center position, render like compartment field color, node shape, reaction field color. And so for render, we support 3 types of color, like the decimal RGB like this, or with the name white, right, black, or a hex string. So you can set, then you can set the color of the compartment, no field color, text font color, and so on and so forth, as you want. Store GRN or only metabolic networks. So SBML, this is not a question for me, right? Okay. Okay, and then, so for the model I have shown you, that the real model glycolysis, you can use SBML diagrams to change the visualization as you want. For example, in this example, you can change the node shape to rounded ones, and you can also change the text outside the nodes. And so because SBML diagrams can do this repeated things, for example, all the nodes are in rounded, then you can just use a function of it to do that and set all the text to the right of the node. It also has interface to SBGN, so like the complex node or complex centroid, like this. Or it also supports the gradient. Like this node is from white to gray, from left to right. So SBML diagrams also has interface to color styles, so you can set different color styles and store it in JSON files. For example, this one you can see you can set the style, name, and compartment field color, border color, species field color, border color, reaction line color, line field color, line point color, and also so on and so forth. Then with the same model, you can have a color gallery of it, like different types, and then store those color styles and for later use with other models. So we also have a simple animation for this that you can see how it goes. So you can see how the simulation goes. So you might understand this process more clearly by this visualization. Okay, so if you're interested, then you can do a pip install for this package to see some visualization of your models, or improve, edit, or model it, and then save all the information for later visualization use. And everything is online. Now I'm going to show you a short demo about this, just to repeat some examples with real demo. So, so this is the model I have shown you. The Jenna-Wolf glycolysis model. You can just read this file and then draw. Then you can see this example. You can also modify this model to a different visualization style with a function create circle node. Also, you can do interface to SBGN by generate some complex nodes, or like this node with the gradient from white to gray. Okay, so that's all I want to show you. Thank you for your attention.
Let me just quickly show you it running on Colab. So I just did a quick one here while Lynn was speaking. And I just created a new Colab notebook. I loaded in the Tellurium, etc. I then where is it here? Sorry. I then pip installed SBML diagrams. Okay, as you would before. This is what happens when you don't do the -Q flag. You get all this sort of stuff, which is not important really. Then I imported Tellurium. Then I created a quick model. Okay, so this is actually a branch model. I then imported SBML diagrams. I then loaded the model by just asking for the SBML. So SBML diagrams has an auto layout feature, whereas it's not doesn't always work, but we're revisiting that. In this case, it did okay. So I can ask it to do an auto layout because the raw SBML, this one here, has no layout information in it at all. So if I didn't do, let me just comment that out first. Okay, actually, let me bring the draw up to here. There's no point having it down there. And if I run this, it sort of gives me a random, picture. If I run it again, I get another one, right? So because it's got no layout information, it just randomizes the positions. However, if I ask it to attempt an auto layout, it gives a much better looking network. Okay. The glycolysis one that you showed, that was manual. Just properly.
Audio. Oh, there you are again.
Yeah.
I
That would
missed hearing you for a couple seconds. Now you're back.
Oh, yeah, back. Yeah. So so we're continuing to develop this. This is just version 1. The idea being that every model could have attached to it the information on the layout for the network itself. Because if I now, I'll do what. Let me see. How do I, Jin? How do I get the SBML? Is it? Oh no, it's export, isn't it? Jen? Hello?
Yes.
I think it's export, right? Yeah.
Yes, let me double check.
It is.
Yes.
So if you do export, you get the SBML that was in the original, but now if you scroll down, you see this whole load of stuff here, which is all about the layout, the colors and everything. And so the idea is that it'd be nice if all the SBML models had this information in it as well, so that you could also draw the thing too. So anyway, that's just.
I will say that many of the models on BioModels have layout information, but by no means all. But you may well find one that
good.
has layout information in it. Yeah,
that's nice. Yeah. Okay. The other thing I can add is Adele. I don't know if he's on the call here, but he was on this morning. He's writing a cross-platform layout app, so you can actually draw the networks and export them as layout and render. So we'll see how that goes. Okay, I think that's it for me. That's just for the addition to what Jin said. Any questions on that? No.
Someone asks how to use the SVGN bit.
Oh yeah, Jen, maybe you can answer that.
So for SPGN, usually, like I have shown you that so for SGN, usually they have some complex nodes or complex centroids. So basically, those are some shapes. So you can add some shapes, for example, add ellipse or something like that as you want. Or if you want to add gradient, you do like set node field linear gradient. We also have radial gradient, so you can set the different types of gradient with colors, with different types of gradient like that.
I mean, I
Is
think
there any sort of auto SVGN thing?
Oh yeah, this is going to ask. Yeah,
Oh, froze.
Jim, you froze.
So have you seen? So I have tried to explain the SPGN.
But is there a more so at the moment you have to manually specify everything, right?
Yes.
Is there any way to say, I want a dimer, and it draws a dimer?
No, not right now.
Okay, would that be worthwhile having?
Okay.
Depends on how many people need SVGN versions of things. Yeah.
Yeah, I think they're probably worth adding an extra layer on top because it is a bit, the API is pretty raw, so you have to build everything up yourself.
Low level, yeah.
Yeah, low level, but there could be a case for having another layer on top that makes it much easier to build, specific SPGN diagrams. For example, if you had a tetramer and it had a phosphorylation site on the second subunit, it'd be nice to be able to have one line that just specifies, create me a tetramer with a phosphorylation site on, subunit 2. And it would just create it. That would be probably, is that the kind of thing you're thinking of, the questionnaire? Adriano? Probably something like that, right?
Yeah, so right now the APIs are very raw. So if you're interested in using SBML diagrams to interface with SPGN, you need to add different shapes and add complex situations by yourself. But if we have the higher layer APIs later, then it'd be more useful probably.
Yeah, I mean, we started working on version 2, so this is definitely something that could be. I realized that early on that the API was a bit raw. Nobody in their right mind would probably do it. We needed a higher level API, so we probably will do something like that. Anyway, yeah, thanks for that question. Okay, in that case, we can go on to the next bit, which is my bit, I think. So let's go to this one. So this one is
So Herbert, I'm going to stop this recording and start a new one, I think.
Oh yeah, please, yeah.
So